Hello, this is a short 5 minute or less video. Some of you have been asking about, first of all, Artemis and non-homologous end joining, and also a little bit about Dr. Mukherjee's non-coding regulatory RNA. Let's start with, here's the outline over here for you to follow along on the right. So Artemis, let's just make this quick and dirty. The point that was made about Artemis is that Artemis and many other nucleases, that is, proteins and protein complexes that cleave DNA. Artemis is interesting because it can cleave in various configurations depending on what is needed. For example, let's suppose we get a non-homologous end joining operation in play here where we've got a double-stranded break. We know that the first thing that we need to do is stabilize those breaks with Q or Cu. And then the role of Artemis is to optimize how this non-homologous end joining is going to go by proceeding in various configurations. We could do a 5' endonuclease, a 3' endonuclease on either or both strands. We can make crazy hairpins and do different kinds of removals. And to better illustrate this, I couldn't find anything better than this paper. So examine here exactly what we said. We've got a double-stranded break here. First thing we want to do, if we're going to go down the non-homologous end joining pathway as opposed to homologous recombination or others, is to perform an end protection by Cu7080. This recruits then a complex of players, including the nuclease Artemis. And what you can see here, if I go down a little bit, right here, it says, and I hope that you can read this, Artemis possesses a unique activity, and so it's got this ability to hydrolyze DNA and RNA in various configurations, and that's really the point that Dr. Martin was making. And so if we go down here, what are those configurations? As I mentioned over here, once we get that stabilizing by Cu binding, now we're in the non-NHEJ type of process, and any number of things can happen. And look at Artemis's role: hairpin opening, 3' endonuclease, 5' endonuclease, and removal of adducts that could be in play prior to Artemis cleavage. And then what we get and the output is some kind of optimized and completed repair. So I hope that takes care of a couple of questions about what is the importance of Artemis in non-homologous end joining. Let's move on and go to a couple of muddy points about non-coding regulatory RNAs from Dr. Mukherjee. The biogenesis, mechanism of action, and functions of these. Hey, look, he said that the biogenesis of these is very complicated, and nobody, even he, could recite all of it. So let's keep it simple. These are either transcribed as is, or they are transcribed and processed out of something else as introns. And so regulatory RNAs can be just made de novo, but they can also arise from cut out intronic portions of other transcripts. So that's where they come from in a simplest form for biogenesis. Number two, what is their mechanism of action? Well, in general, there's two ways that these things work. First of all, they will bind, and this is straight Watson-Crick type of or Chargaff base pairing. And in this case, this can be sent to a RISC complex and then degraded. Alternatively, the target mRNA can be bound by a non-coding regulatory RNA and for some time be unavailable for translation. Some of the most effective of these are up in the 5' untranslated region, so then ribosomes and things like that can't get on, and EIF factors to initiate translation can't get on. But remember that these are RNA protein complexes. We really ought to call DNA and RNA DNP and RNP because they never exist naked. They are always bound by protein complexes. Many of these are deposited by the RNA polymerase C-terminal domain, by the way. Once these factors get on here, then it depends on what kind of non-coding regular RNA is being applied and where it's bound to, where you can get decoys, scaffolds, guides, and enhancer action, as Dr. Mukherjee indicated. And so for you as clinicians, what kind of tools as a scientist helping you to be the best clinician you can be, what can we give you? What kind of tools are you going to give your patients? Not only right when you get out of med school, but certainly into your career, this is going to be very, very common. So let's consider RNA as therapy, but let's talk about, before we get to using CFTR as our target disease that we want to treat, let's step back a little bit and talk about, wow, some of the problems. Why is it not quite ready for prime time? Well, consider this blood vessel here. So let's say that we have done an IV injection with our RNA therapeutic. Recall from the previous slide that we probably need to coat it with something, proteins or other things, to protect it along its journey through the vasculature here. But we've done an IV solution here, and we've dripped in our RNA therapeutic. Consider that it comes out of the capillary bed. That's some fenestrated endothelium there. Now it's in the extracellular space, and it's getting ready to be delivered to our target cell. And let's assume we don't want it to bind this non-target cell. How would that work? Well, right here is a big black box. How are you going to get this across the membrane? Very often, these are complexed with lipids and other things that allow, almost in a steroid type of way, to get this guy in here. So, yeah, lipid delivery is one thing that was mentioned in the lectures, and a very promising technique. Once the RNA gets in here, let's say your therapeutic is to transcribe something, then hopefully it will find its way to the ribosome and be translated into your protein of interest. There's a lot of sort of things in a black box here that need to be worked out. Also, we still have not solved our problem of non-target types of cells, and this can be very dangerous. So we still have a lot of work to do here, but you can see that crossing membranes is one problem. Another problem is immune activation. Sometimes these RNA and lipid complexes can cause an immunogenic type of situation that is really unwanted. Third, our targeting. In this case, I put anatomy. Remember, if our target cells are, say, somewhere in the brain, we could do an intrathecal type of injection. Let's say that they're somewhere else. We can use our knowledge of anatomical barriers and openings and other places to try to really deliver this in a much more targeted fashion. So we've got a biochemical way to get some targeting, but let's don't also forget anatomy. We can use the anatomy to help us out. Once we do this, what do we want to do? Generally, we want to off a gene, and I mean off a gene by turning it off. So this would be in the previous slides where we create a double-stranded form of something and we activate pathways with RISC and Argonaut that destroy that or otherwise inhibit its translation. A second way we can think about doing this was the example I gave here, where we're supplying an mRNA that we would like to get translated into a protein. So we'll on a gene. Third, we could, by this on a gene approach, actually immunize somebody to make an antigen, and we'd have to get access for this to the immune system so that we could get macrophages and these other players to then go ahead and activate our acquired immune system and perform an immunization. So turn a gene off, inhibit transcription and inhibit translation, or promote transcriptional degradation or RNA degradation. We can turn on a gene, get it to be translated, and we can do that in a specific way maybe and immunize folks. And so here we go. Here's our last bit. Is the example given in class was, how do we fix cystic fibrosis, let's say? Autosomal recessive, 2 bad copies of CFTR. So that's the sort of pathogenesis. So the simplest way would be to perhaps do the on a gene approach, where we're asking, okay, let's supply the mRNA that is not here because we have some kind of DNA mutation in at least 2 copies of the CFTR. The question here that you should grapple with is, for durability of that type of approach, do we want a transient or do we want a stable type of approach? What I mean is, if we have to continually pump mRNA here to get our CFTR made and get our channel out here, that is obviously one approach. Another approach would be if we could get this somehow in the nucleus and just give us that one functioning copy, then we're going to be okay. So I mentioned durability, and let's go ahead while we're at it, talk about targeting. In this case, the lung, we would probably prefer a nebulized approach. So intranasal, I won't draw somebody's head, that'll be crazy, but intranasal or intrathoracic would be a way to target lung-only cells. We still don't have any protection against perhaps non-targeted cells, but in any case, restoring CFTR function by an mRNA or an RNA type of therapeutic in a directed fashion would certainly be possible. Hope you enjoyed the video, and I look forward to perhaps doing a few more. Thank you for watching.